Hi, so my work is on Lyme disease pathogenesis, and so Borrelia Burgdorferi is depicted right here. So it's the bacterial pathogen that causes Lyme disease, and it's transmitted through the bite of an infected tick. So those ticks are right here. You can see that they're actually pretty small, and so sometimes you might not realize that you have this small tick on you, and they're slow feeders, so they feed up to a week, and they're maintained, the bacteria is maintained in the environment through a small mammal population. So there's a naive tick, and if it feeds on an animal in the environment that has this pathogen, then the bacteria will go into the tick, and then once the tick molts into its next life form, that bacteria is still there, and then it's able to reinfect other small mammals in its life cycle, and then therefore it keeps the bacteria in the environment. But what can also happen is if you're out hiking, then maybe a tick can encounter one, and in that instance, the bacteria can go into your own body. And so when that happens, typically you develop this bullseye-like rash, which I don't know if you can see there, but it looks like a bullseye. It's called an erythema migrans. It's actually your body responding to all of these bacteria that are now swimming away from that tick bite and going everywhere else in the body. And so if you leave this untreated, you're going to develop flu-like symptoms. But then if you continue to leave it untreated, you can develop more severe conditions such as arthritis and carditis and various neurological problems. So it can be a very debilitating disease. So what our lab is interested in is how can it live in all of these different places? I mean, it can live inside of a tick, inside of a mouse, in humans, in dogs. And it's a unique pathogen. It doesn't meet the normal dogma as other bacteria go. And we don't really understand a lot about it. And specifically, we're interested in understanding what's going on inside of the human host, and how can we model that and figure out what are the genes that are involved in the ability of this bacteria to survive inside of humans. And so we've taken a couple very large global screens to look at that. And if you come to my poster, I can tell you about how we did that specifically. But we just asked this simple question and said, what genes are turned on in the bacteria inside of a mammal? And then from that, we can learn a lot more about its pathogenesis. But when we did this screen, we actually found we found genes that were turned on, but we found all of these other elements in the bacteria that were important, that we believe to be regulatory factors. And so, come to my poster, I'll explain to you more in depth what that is. So this is what my work is. So we found all these genes, we found all these other interesting elements, and now what I'm doing is actually I have bioluminescent bacteria, and I can measure these bacteria inside of living animals. So I have a mouse model. I can inject my bacteria inside of these mice, and I can actually measure the expression of all of these things, all these genes that we're finding. So that's what I'm doing with my work.
Right on time.